In this video, I will be explaining Figure 2 from the research paper, Profilin reduces aggregation and phase separation of Huntingtin N-terminal fragments by preferentially binding to soluble monomers and oligomers. The focus of this paper was to understand the mechanism of suppression of Huntingtin N-terminal fragment aggregation, with hopes of future applications of therapeutic approaches for Huntington's disease. Before I begin with the figure, I would like to provide some background information, starting with the Htt-NTF protein, the protein's relationship to Huntington's disease, and finally the concept of polyphasic linkage applied to Profilin and Htt-NTF. Huntingtin N-terminal fragments, which I will refer to as HTT, are made up of three main parts: the N-terminal stretch, a polyglutamine tract, and a C-terminal stretch that includes two polyproline segments. HTTs with longer polyglutamine tracts form toxic aggregates that are associated with Huntington's disease. Huntington's disease is a neurological disorder that affects the neurons in the brain. Age of onset of this disease and the severity of its symptoms are inversely related to the length of the polyglutamine segment in HTT. This is because HTT proteins with extra long polyglutamine regions form insoluble aggregates in the neurons. There are three phases in HTT aggregation. The M phase is made up of soluble monomers and oligomers, the S phase made of soluble spheres, and the F phase made of linear fibrils that are insoluble. The three phases are separated by distinct saturation concentrations or concentration thresholds. For example, when the concentration of aggregates in the M phase reaches a certain point, aggregates of the S phase begin to form. However, concentration thresholds can change through binding of profilin, an abundant protein in cells, to HTT aggregates. This results in reduction in aggregation and brings us to the concept of polyphasic linkage. Polyphasic linkage demonstrates the impact of ligands on saturation concentrations. In the case of profilin and HTT aggregates, profilin is the ligand that binds to the Huntingtin aggregate. Profilin also prefers to bind to the M phase species, resulting in a change in concentration threshold between M and S phase. This is all summed up in Figure 2, which shows the phase behavior of Huntingtin aggregates and the influence of polyphasic linkage of profilin. Figure 2a shows the behavior of HTT in the three phases. The M phase is colored red and shows the soluble monomers and oligomers. The S phase is colored blue and shows the spherical aggregates. The F phase is colored green and shows the long insoluble fibrils. Along the horizontal axis, gray lines labeled CS and CF indicate the phase boundaries relative to the total concentration of monomer equivalent aggregates. For example, when aggregates present in the S phase reaches the concentration of CF, aggregates of the F phase begin to form. Figure 2b shows the influence of profilin binding on the CS concentration threshold between M phase and S phase. In the first scenario, no profilin is present and the phase boundary is shown as a solid gray line. Scenario 2 shows profilin, the black dots, preferentially binding to the M phase and increasing the concentration threshold. Scenario 3 shows profilin binding to the S phase and decreasing the concentration threshold. Scenario 4 shows profilin binding equally to both phases and no shift in the concentration threshold is observed. In summary, profilin affects the toxicity of Huntingtin aggregates by its influence on concentration thresholds in aggregate formation. Understanding of this regulation of aggregate phase behavior in cells can lead to development of ligands that can be used against aggregation-prone proteins in Huntington's and other diseases in the future.